Hello there. This is Linfa Wang, and I'm the professional director from the program in emerging infectious disease at the Duke National University Singapore Medical School in Singapore. It's my pleasure to have this opportunity to present you on the topic of challenges and the role of serology in the next phase of COVID-19 responses. So this is a brief outline. I hope most of the audience will be very familiar with the COVID-19 outbreak. So I would really go through very briefly, and then maybe a few slides on the disease and virology. And then I will present the challenges I think are there for the next phase of the outbreak. But the bulk of the talk is really on the role of serology. So in terms of the outbreak, it's been going on for 6 months now. It started in December 2019. And as of yesterday, we have already over 7 million infections and close to 430 deaths. And I have to say, I have been working in this field for the last 25 years, and I was in Wuhan in January 2020. But I even myself would never predict it. There's two things really sort of caught us by surprise. One is the speed of spread and the number of infected people all around the world. Secondly, as you can see, is the dark, the color that's the epicenter. And nobody would have predicted, right? So these are the areas, like in USA, can be affected so badly. In terms of the major events, we already know that, early December might be the starting of circulating the virus in Wuhan. And by January 7th, China CDC eliminated to say it's not SARS, it's not avian flu, but it's a novel coronavirus. And the first death was registered January 11th. And then in Thailand, the first export cases basically from China. And the WHO declared a global health emergency on 30th of Jan. And, the virus was named as SARS-CoV-2. And then March 11th was WHO declared as a pandemic. On this time sort of scale, there are two important events, for scientists. I think it's really important for scientists like myself. One is actually that on January 11th or 12th is the first genome sequence become publicly available. In this day and age, whether you want develop diagnosis or vaccine sequences is absolutely essential. And then another important day is on January 20th, because that's the date basically that the Chinese authority announced that there's human-to-human transmission. Once the virus is capable or known to have a human-to-human transmission, then the whole world take it much more seriously. Early on, there was a debate about this new outbreak. Is it SARS or not SARS? I always say it depends on what you want to focus. If you focus on the disease, then SARS is severe acute respiratory syndrome. And we know for COVID-19 now, more and more data comes out, is the majority of patients present with mild disease, and now there's a high proportion is asymptomatic. So in this regard, then I think the original claim is right, that it's not SARS. But if you look at the virus, and the virus, the genome is 80% identical to the SARS coronavirus. And in virus taxonomy, we have 4 levels. We go from the, orders to families to genus and to species. So this virus is in the same species as SARS coronavirus, and the species name is SARS-related coronavirus. So I always use the analogy for humans, right? We are all one species and different strain, so different ethnic background. So in this case, SARS and COVID-19 are the same virus, just different strain. Most people are familiar now with this computer-generated image. So it's a beautiful virus with the spikes on the surface. The name comes from this crown-like. If you do a section cut through the virus, and then a spike protein present on the ring, it's like a crown. So that's why it's called coronavirus. And the key proteins on the membrane surface is this spike protein, which is responsible for receptor binding and the fusion with the host membrane. And the genome is large, so it codes for the spike protein in green, and then the nucleic capsid in brown, and then the E and M protein. So these are the 4 structure proteins. and the others are called ORFs, so they are non-structural proteins. The genome is huge. RNA virus usually have small genomes because the RNA polymerase has a low fidelity, so that usually you could not maintain a large genome, but coronavirus is exception. So the genome size is around 30 kilobase, and it's the largest genome of any known RNA virus. SARS-CoV-2 seems to be also genetically more stable than the other two coronavirus, the zoonotic coronavirus, SARS coronavirus, and the MERS coronavirus. It uses the same receptor as SARS coronavirus, the ACE2, on human cells, but with a high affinity. So this has been, speculated, maybe explain why the COVID-19 virus is more transmissible than the SARS virus. And in terms of origin, we don't know for sure yet, but most likely it's another bat-borne virus because a group in China already discovered there's a bat virus, which is 96% overall genome identity to this SARS coronavirus 2. And the question whether there's an intermediate host, we don't know, but a virus has been discovered in pangolin, So again, it's a COVID-19 related virus. The overall genome identity is not as high as the bat, but it's being discovered that actually the key receptor binding domain of the spike protein is more related than the bat virus. So this is shown in the next slide. So this is the paper, in Nature published a few weeks ago now. And the phylogeny on the right is that in the pink box, basically these are all COVID-19 related virus. And the human virus is the main one. And the red dot marked the virus that discovered in different pangolin studies. But the red arrow indicates this is the virus coming from bats. So the bat virus is still more related to the human virus than the pangolin virus. Interestingly, so for those, of course, you could not read the Chinese, I apologize, but this is the menu from the Huanan seafood market, was very famous because the early cases seems to be all linked to that seafood market. And so this is the menu in Chinese. As you can see, there's a lots of animals or meats are sold, but not pangolin, okay? So the red box indicates that actually it's a civet. So civets we know is a highly susceptible, responsible for the transmission of SARS coronavirus in 2002, 2003. So it's an intermediate host. But for COVID-19, we still don't know whether an intermediate animal host is involved and what is the animal. So we still don't know. So in terms of challenges, there are many, but I want to focus on three challenges, really, to contrast COVID-19 to SARS. For SARS coronavirus outbreak, it's lasted totally around 6 months and with just over 1,000 infections and 774 deaths. The virus, basically, or the pandemic, was contained or eradicated by July 2003. Now, in contrast, we know that, this is an old number. So we already have over 7 million infections and about 430, deaths already. So the chance of containment or eradication is slim, if it's not zero, right? So what we need now, the challenge number one is exit strategy. That's we did not face during SARS outbreak. Challenge number two is that obviously vaccine will be a very important part of this effective exit strategy, but we're still a long way away from a licensed human vaccine. But before even the vaccine comes on market, we really need to get our tools and knowledge to really lift our game, is because the correlation of protection is still poorly understood, and also the longevity of protective immunity will not be known until a few years later. Right? We cannot start longevity without actually let a recovered patient run through the process. But maybe there is a little bit sort of good news. If we think SARS coronavirus and the COVID-19 are so close related virus, if the human immune response are similar as well, then we have some good news. So our group just published this paper a few weeks ago. We checked the patient sera from SARS survivors in Singapore. So these are the patients that infected 17 years ago, and we take the blood from donors and we compare their neutralizing activities, with the serum that we have archived, so that in 2003, and we define them as convalescent serum less than 1 year old, and then we have convalescent serum, in the table, as you can see, 9 years, 14 years, and 17 years. So if you focus on the 9 to 17 years and to the SARS coronavirus, as you can see, that majority of these survivors still have quite high neutralizing antibodies. So that's hopefully is a little bit good news if the COVID-19 immune response resembles that of SARS patients. Challenge number 3 to do with animals. As I said, we believe it's most likely a bat virus, but the intermediate host is not known, right, whether there's intermediate. Then, different from SARS, SARS, as I said, is relatively contained and did not spread too far. But for COVID-19 now, we have this concern about this spill back or reverse zoonotic transmission. So we think initially the virus was animal to human jump, but now there are over 7 million people involved, infected. Some of these infected humans will have close contact with animals. So there's a chance for this transmission from human to animal, what we call spillback or reverse transmission. And this is a report just came out of Science. So for those of you who are not aware that, in the mink farms in the Netherlands, now there is a pretty bad outbreak of COVID-19. And this is clear spillback or reverse zoonotic transmission. And to a certain degree, it's not a surprise because mink is related to, ferrets, right? And we know that ferrets are quite susceptible to this virus and also to flu, So this is the space we need to watch out. The other concern is that it may spill back to bats, but at a different geographic location. So, for example, in North America, we are already concerned whether the human infection can result in a spill back of this virus into bat populations and then really form a new reservoir in a different geographic location. So this is sort of the bulk of my talk, is that I have been, focused on serology since day one, as soon as we heard the COVID-19 outbreak, because I believe that serology in the late phase of the COVID-19 response will play a major role. So here I just listed some of these, challenges we're facing, right? You heard of this, herd immunity, how much we have to get in the herd immunity before we can stop the transmission. Some countries are considering, exit strategy is to issue immunity passport. As I said, the longevity of the protect immunity, we still don't know. And convalescent plasma is being used as a therapy, but we don't know, whether any patient's convalescent plasma is good enough. And then once we have a vaccine, then the efficacy at both individual and population level. And then we have the animals. So the top 5 are human really applications in red. And the next 2 dot points are in blue are to do with animals, so whether we want to search for natural reservoir or intermediate host or monitor the risk of reverse zoonotic transmission. In all of these areas, serology will play a very important role, but we need to really go beyond just detect antibodies. What we need for the red 5 dot points is really have a serology which can detect functional antibodies or neutralizing antibody. For the blue dot point for animal studies, then what we need is a serology test, which is species independent. Basically, we don't know which animals we're going to test, right? And so we don't want to develop a specific serology for each animal that we want to test. So here is, again, this diagram of the virus and the genome structure. When a virus infects a host, whether it's animal or human, it will induce lots of antibodies, hundreds, if not thousands. And for large viruses like coronaviruses, of course, the antibody will be directed to all sorts of different proteins and different domains. But these, what we call, are binding antibodies, right? As I said, in the previous slides, for most of the next phase response, we want to know the neutralizing antibodies. Unfortunately, neutralizing antibodies is always going to be a minority. So for coronavirus, that may be 1 to 5%. And we know that the spike protein is involved in virus, binding to the receptor and the fusion with the cell membrane. So this is the main target. The green protein, the green one spike, is the main target. And within the spike, there's a domain called the receptor binding domain, now is highlighted in red bar. So this is a very important domain because that's where it makes the interaction with the human ACE2 receptor. And it's even more important in terms of detect neutralizing antibodies. So currently, the gold standard of detecting neutralizing antibodies is using live virus, okay? So this is a diagram on the left. You have a virus, docking onto the receptor. In this case, it's ACE2, and then start this entry process followed by replication and infection. In the presence of neutralizing antibody on the right of this diagram, what the neutralizing antibody does is that they can block that docking and then prevent infection. So the advantage is that it's highly specific, okay? The disadvantage is that for COVID-19, it's a biosafety level 3 pathogen, so you have to have a BSS-3 before you can do it. And it's tedious and slow, it takes 3 to 5 days. And it requires highly skilled staff because the read out is not as simple. So you need not only a facility, you need really special equipment, but also that the readout, for example, this is the true photo we have taken on the cell, is that in the presence of neutralizing antibodies, the cell is healthy, but without neutralizing antibody, it causes a cytopathic effect of CPE. But for untrained eyes, you look very similar. It looks very similar. So to solve that, really to address these challenges, we developed a surrogate virus neutralization test. So this really addresses both challenges by determining these neutralizing antibodies, but also in a species-independent manner. Okay. So the invention itself, on the theoretical level, is very, very simple. So this is a different version of the diagram I just showed you. So this is now the coronavirus here, and this is the spike protein on the left in blue, and interact with the ACE2 receptor in purple. And then on the right side, if you have the neutralizing antibodies in the dark brown color, that will bind to the RBD, receptor binding domain of the spike protein, which will prevent the binding to the ACE2 and achieve a neutralization. So what we did is basically a biochemical simulation. We do away with live virus and we don't need a cell. So what we did is using protein engineering, basically. So both the spike protein and the ACE2 are membrane proteins. So in order to do biochemical simulation, we have to engineer a protein which is not membrane bound and soluble. So that's what's on the right, okay? The right and the bottom left. So what we have now is we only express the blue head, the receptor binding domain, rather than the whole spike protein. To make the life is even easier, we chemically conjugate with the horseradish peroxidase. That's the enzyme ubiquitously used in ELISA assays. So we chemically conjugate this. So this is indicated in yellow. And so we have HRP IBD conjugate. At the bottom, the line is the bottom of the ELISA plate well. And as you can see, we coated with the ACE2, but now the ACE2 is engineered, so we remove the transmembrane domain, so it's a soluble protein as well. So in panel B on the bottom left, so that if you have an HRP IBD and you bind to different amounts onto the ACE2, and then you put a substrate, you see a color. So it's a dose dependent. On the right of panel B, in the presence of neutralizing antibody now, it will bind to the IBD domain and block the binding of the ACE2. So basically what you're doing is that reduce or eliminate the color reaction. So here is the real data. So in panel C is that we use the 3 proteins, the nucleic capsid protein N and the S1, half of the spike protein, and then the RBD. So as you can see, the N protein was used as a negative control because it does not bind to ACE2. As expected, the blue is the S1 and the red dot is the RBD. So RBD gave us the best binding in a dose-dependent manner. In panel D now, we tried this neutralization. So this is a surrogate neutralization. Everything is done in an ELISA plate where no live virus, no cells, no BSS3. So as you can see, patient 1 and 2 in red and blue give a beautiful inhibition curve, dose dependent again. When you dilute it out, the inhibition is gone, whereas the 2 negatives has no inhibition. So we were really excited when we saw this data, and we thought ACE2 is the same receptor used by both COVID-19. So this is panel C and D is COVID-19, but it should also work with SARS. So in panel E, we did the exact same thing. Now it's a SARS coronavirus RBD in red as well. And you can see a beautiful dose-dependent binding. In panel F, we did a serum neutralization and the 2 positive SARS patient and 2 negative. As you can see, again, beautifully dose-dependent and very specific neutralization. So as I said, we want to develop not only a surrogate neutralization test to measure human antibodies, but also to measure animal antibodies in a species-independent manner. Can we do it? So these are the data. So on the left is COVID-19. So for COVID-19, we don't have infected animal serum in our hands, so we use immunized serum. So these are rabbit and mouse immunized with the RBD domain. As you can see, the negative control, there was no inhibition, and the positives are beautiful. The inhibition is very high, close to 100%, and can be diluted out, of course, in a dose-dependent manner. In panel B now, this is the SARS serum. So I do have some ferrets infected with SARS. So this is a natural infection, and also rabbits, immunized with a whole inactivated virus, the whole SARS virus. That was done, 17 years ago. Again, beautiful inhibition and none with the negative control. So basically, we have demonstrated in 3 different animal species and 2 viruses in a totally species-independent manner. So the same assay can be used basically to test any serum in a very specific and sensitive way. Now, this is important because we already knew that for SARS coronavirus 2, that the human-to-animal transmission has already happened in cats, dogs, tigers, and minks. And we know natural infection happens in the wild with bats, pangolins, and the civets. Okay, so I can envisage many more animals are susceptible, and we need to really do large-scale and multi-geographic location serology surveys to really find out, COVID-19 related virus, how widely distributed, and whether any of these animals could be the intermediate host. These informations are important to prevent future outbreaks. So when you have a surrogate virus neutralization and we want to know how specific it is, so we did some cross-reactivity. And as you can see from in panel C, you go from right to left. So panel C, we have most serum which does not inhibit or does not neutralize this in the surrogate virus neutralization assay. The 229E and then the 63 does not work. OC43 does not. SARS is interesting. As I said, we have 2 groups of serum, right? Convalescent serum from SARS survivors in Singapore. We have this in 2003, less than a year convalescent, and we have 2020, more than 17 years of convalescent. Interestingly, that the 17-year serum is more cross-reactive. So they cross-neutralize the COVID-19 virus. To make sure that these are not SARS survivors have been exposed to COVID-19, which is very unlikely because in Singapore, the population's prevalence of infection is less than 0.1%. So for somebody to have a SARS 17 years ago and exposed to COVID-19. But we still want to make sure that's not the case. So this is what we did. So in panel D, so this is the less than 1 year convalescent serum. So now we are doing this surrogate virus neutralization for both COVID-19 and SARS side by side. So we should be able to tell, which is it more SARS or more COVID-19. So clearly that, in panel D, that they have very high inhibitions on SARS surrogate neutralization, but not on the COVID-19, which is in blue. But in panel E, that 17 years later, as you can see, overall, the gray dot are the SARS surrogate virus neutralization. So the neutralizing antibody to the SARS virus has dropped a little bit 17 years later, but still there. And then, but some individuals have increased now COVID-19 cross neutralization. So it's a very interesting observation. The mechanism we don't know yet, but I think more research needs to be done. Okay, so another question reviews really ask about is that you claim this is a surrogate virus neutralization, and how well does it correlate with live virus neutralization, right? So currently we have two live virus neutralization. One is using this COVID-19 virus and has to be done in BSS3. So this is the correlation curve, as you can see on the x-axis is the conventional virus neutralization, the micro neutralization test we call CVNT. On the y-axis is our surrogate virus neutralization. And as you can see, the correlation is pretty good. R squared is 0.86 approximately. We also did this with pseudovirus. So the pseudovirus neutralization, and we call PVNT on the x-axis, and the y is still SVNT, and the R squared is around 0.83. So similar, not as good maybe as the live virus, but it's very similar. Okay. So we went ahead and used human series both in Singapore and China. So we had a 200 negative series in blue, and the red, 170 something samples in Singapore and 50 in China. As you can see, panel D and E, that the specificity in both cases is 100%, and the sensitivity varies from 98 to 99%. So I have to stress that these are convalescent human series, a PCR confirmed, okay? So as some of you may know in the audience that for COVID-19 patients, that not everyone seroconvert. So there are people get infected but don't seroconvert, okay? So another question is to say, now your surrogate virus neutralization is a good idea, but you're only measuring antibodies targeted at RBD. So are you gonna miss non-RBD binding neutralizing antibodies? So specifically, there are several scenarios. One is that in the S protein, you have the S2 domain, for example. From the SARS studies, they have demonstrated some of the S2 antibody can also play a role in neutralization. And recently, the neuropilin has been identified as a potential, a co-receptor. So where the co-receptor play a role, where the IBD will interact with that. And also that, for in general, virus neutralization can happen post-attachment or post-entry. So I would say the answer to this question is yes, in theory, but on the operational level, if you're measuring neutralizing antibody using viral E6, then none of these will play a major role. So just a few days ago, there's a Science Translational Medicine paper, journal published a paper. They did a very detailed, really immunization studies, and then they found that the RBD domain, not the S2, really generates strong neutralizing antibodies. And our own studies also demonstrate that. So this is another correlation. So this is a CVNT correlate with SVNT. I have already shown in the data. And now one is the CVNT correlate with the pseudovirus. So the pseudovirus used the full-length spike protein, whereas our surrogate virus neutralization used the IBD. If the anti-S2 antibody plays a major role, you will see the difference, right? The pseudovirus will perform much better in correlation with the live virus, but it's not. As you can see, the correlation R squared is 0.86, and here is 0.77. So if anything, that our RBD actually based surrogate virus neutralization correlates with the live virus neutralization better than the pseudovirus, which is based on the full-length spike protein. So that's also indicate that IBD really is the major neutralization sort of a domain, and the correlation at the operation level is really good. The other question is, can antibody testing protect immunity? That's a really in the news all the time. So basically, you can split into two questions. One is that binding antibodies, does it equal to neutralizing antibodies? And we know it doesn't. Secondly, is that neutralizing antibody level, does it equal to protect immunity? And we would say that, approximately you can use that as an estimation. A few days ago, last week now, so when the Moderna vaccine, announced the press release, Tony Fauci made this statement to say, the exciting thing is really they did not just show binding antibody, but they show neutralizing antibody. Then he went on to say that, that will predict, a protect immunity, and it's a reasonable assumption that neutralizing antibody will equal to protect immunity. Okay, so that's what we try to demonstrate. Surrogate virus neutralization differs from all the other antibody tests. Even if you use IBD as antigen in a commercial test, whether it's ELISA or rapid test, you are detect binding antibody. So in order to differentiate the normal ELISA and our SBNT, so we did this study. So we use mouse monoclonal antibody, 4 of them. On the left is ELISA. Panel A is binding. As you can see, all 4 monoclonal antibodies have a good binding and a dose dependent. Panel B is the surrogate virus neutralization inhibition. So as you can see, the red one, for example, has that monoclonal has a very strong binding in panel A. It's a neutralization negative in our test. So our test can really differentiate between binding and the neutralizing antibody, and we are truly measuring neutralizing antibody. And this is a human monoclonal antibodies, again, 4 of them. And then this time, as in panel G, as you can see, this yellow monoclonal has a strong binding. And on the panel H, in neutralization, it's negative. One thing we're really surprised, to also observe is that when you put these 2 neutralizing antibody together, so this is the blue and the green. So when you put the blue and the green, you should get an average. So the predicted value is this dotted line in gray. But instead, what we get is the solid line in purple. So what we're seeing is something called the neutralization synergy or neutralization epitope synergy. So when you have antibodies bind to different neutralization epitopes, they can have a synergy, okay? So we think we have seen this for human sera as well. So these are the human sera from Singapore that we functionally divide them into, from ELISA with IBD, we have 4 different groups. So if you go from the bottom to the right first, panel D, which has high IgG and high IgM, as you can see, they have a very strong inhibition and have a titration curve, so really sort of curves upward. And then you have in panel C, you have high IgG, low IgM. As you can see, still very good. Panel A, now you have high IgM and low IgG. The curve inhibition is good, but the titration, the drop is faster. So the curve has a slope, is a sharp slope. And then you go to panel B, which is low IgG and low IgM. It still have a good inhibition from 70 to 90% at low dilution, but when you dilute it out, now you can see it drops very fast, okay? But what I try to emphasize is in panel B. If you use IgM and IgG, so if you use this isotype-specific ELISA, it will all be classified as negative or very weak positive. But in our virus neutralization assay, we're measuring total and also synergy. If there's a synergy, we will see that. So actually the neutralization is still pretty good. Okay, so with that, I'm going to conclude my talk to say now we have developed this surrogate virus neutralization, which offers many advantages over the existing serology, whether it's ELISA or the virus neutralization. It's our SVNT is specific and sensitive. It's safe. You don't need a BSS-3. Very fast 1-hour test. And that too, if you want the low throughput, you can do manually, or high throughput, you can use a machine. And then can be used for both human and animals, as I said, is a species independent. But the key is that on the market right now, this is the only test capable of measuring true neutralizing antibodies without the need for any live materials, virus or cells, and definitely no biocontainment is required. So with that, I'd like to thank you, and I do have a conflict interest declaration because we have a patent on this technology and a commercial kit is being marketed right now. And thank you for your attention.